and they were not able to fertilize. So still, although he still did that experiment, but he still didn't believe. He still thought, maybe this is my experimental error, and sperm are still parasitic. Then in 1824, this is kind of interesting. There were a guy, Prevost and Dumas. These two guys were there, Prevost and Dumas. What they observed, they noted the existence of sperm. They noted existence of sperm only in sexually mature males. And what they found, that aged individuals and sexually immature, or the young immature individual, they didn't have sperm in their semen. So, and they also observed that the sterile mule was devoid of sperms. So this is the first time they thought that sperm actually enter in the egg and they start the process of the fertilization. Then in 1876, this guy Hertwig, he actually demonstrated in the sea urchin egg that sperm actually enter the egg. They enter the egg and their nucleus of the egg and the sperm, they fuse. So sperm, what this guy said, they said sperm enter egg, and once they enter, nucleus formed by a fusion of sperm and egg contributes to the nuclei of new organism. So these guys were basically able to demonstrate that sperm has some role. So far it's okay. It's kind of an interesting historical background. Yeah.
What's the name of the 1829 scientist?
Which one?
The first one.
This one?
Yeah.
Prevost and Dumas. Okay, now. Now we're going to talk about the structure of sperm. Now, the sperm has three major components. In a structure of sperm, there are three major components. The first component is haploid nucleus. The sperm has a haploid nucleus. And then this haploid nucleus, it has a tightly compressed DNA, tightly compressed DNA. Okay. And then the sperm has a propulsion system. Sperm need to move, right? So for that, this propulsion system is to move. It needs to move. And then the sperm need to enter the egg. And if need to enter the egg, it should have the enzyme. And those enzymes are present in a sac of enzymes. So it should have these three things. So sac of enzyme to enter the egg. These three are the main components which a sperm must have. Okay, so how these components are arranged in the egg. So in the egg, or in the sperm, sorry. So this is the structure of the sperm. The sperm has this structure right here. And then this is called sperm head. Okay. In the sperm head, what you have is, you have a nucleus. This is haploid nucleus. And then, right there, there is a sac of enzymes right here under the sperm head. Now, this structure, which is the sac of enzyme, this is called acrosome. Acrosome. So this acrosome. Okay? Now, this is sperm head, essentially this whole process, but this is sperm membrane. Okay. Now, then after the sperm head, there is a area. So this is sperm head right there. Then this area is right there. This is called middle piece. This is called middle piece. Now, in this middle piece, what we have here, right here, we have centriole. How many centriole are present in one cell? 2. When? All the cells have 2 centriole every time? They have 1 centriole. At the time of division, they divide and travel to the other side, right? If you remember your mitosis, right? 1 centriole, prophase, divide, move to the 2 sides. But the sperm has 2 centrioles because the egg will have no centriole. We'll talk about that later when we come to fertilization. But there are 2 centrioles there. So these are the centrioles. 2 centrioles there. And then there are huge number of mitochondria here. These are all mitochondria. These are all mitochondria. They're all mitochondria. Now, then after this area, so this is the middle piece, and then you have a structure like this. It's going like this. Okay? Now this is called, so this was middle piece, now this is called tail, and this is called end piece. End piece. End piece. So we have the end piece there. Okay. Now, then this end piece is actually a flagellum. Okay. And when we say flagellum, so you should have the flagellum proteins also, right? The protein of flagella starts from here like this. And this actually, end piece is actually the flagellar protein. And this protein, which is a flagellar protein, that is called axoneme. So this is axoneme. Okay? So far everyone is okay? Now, so what are these things? So let's talk one by one of everything. There is practically no cytoplasm. So practically, sperms are devoid of cytoplasm. So they don't carry any cytoplasm with them. And they eliminate, when the sperms are formed, that process is called spermatogenesis. Okay? So they eliminate cytoplasm during spermatogenesis. So the spermatogenesis, all the cytoplasm is eliminated. Okay. Now, so this is the sperm membrane. Then you have all these structures here. Okay. Now, what happened? So the acrosome vesicle, acrosome, this acrosome. Okay. Acrosome is a Golgi derived structure. Is from Golgi. Okay. And what it contained? It contained digestive enzymes. It contained digestive enzymes. And what those digestive enzymes are going to do? Those digestive enzymes, they can digest sugars and proteins. They can digest sugar and protein. Now, so it means if it is derived from a Golgi, if you remember your cell biology, so from Golgi what happened? You have the vesicles which are coming out, right? Those vesicles can do exocytosis. So it means that it's a Golgi derived structure, it is contain enzyme, that means it is a modified secretory vesicle. Modified secretory vesicle that is derived from Golgi. That is making sense? Now, what is the purpose? It has digestive enzyme that can digest sugar and protein. It means that these enzymes, they will digest a path through outer egg. So egg has the coverings, right? So the outer egg covering. So if they can digest that path, then they can go through that outer egg covering and reach the egg. That is making sense? So that's why this thing is there. Now, then in most of the species, in the right here, between the acrosome and the nucleus, there is a protein right here. This is called F. This is not F actin here. This is called globular actin. This is globular actin. What is the purpose of actin? Going back to cell biology. What is the purpose of actin?
Cell structure.
Say it again.
Cell structure.
Structure.
Structure, that is true, but give me more right answer. A little bit more. So, G actin, what it does? G is globular, right? It's a monomer. Many monomer of actin come, give a fiber, right? So what happened? This globular actin is there. When this sperm reaches, and how that happens we'll talk in the next coming chapters, right? So when the sperm reaches the egg, at that time, this G actin, globular actin, it can change into F actin like this, and it can form microfilaments. It can form microfilament. And what can they do? They can form a process like this. And how that is formed, we'll talk in great detail later, right? Here just, we are talking about the structure, so I'm giving you what is there, what it does. So we have a G-actin, globular actin, also called G-actin. That globular actin changes to, G-actin changes to F-actin, make a fiber, and that make microfilament, and those microfilament make a process, this is called acrosomal process. Okay, so in most of the species, beneath the acrosome, above the nucleus, there is globular actin. When fertilization is going to take place, this globular actin changes to F-actin, and right here on the top of the head, a process will be made that will be called acrosomal process. So far we remember this. What it does? Acrosomal process, it play a role in sperm egg recognition. How that does, we'll talk when we talk about fertilization and all those things, right? So far it is okay? Everyone is fine here? Now, before I erase it, I go to other topic, other part of this. I would like you to talk to each other so that everything is clear up to here, okay? So just talk to each other about what we did. So then I'll erase it and go to the next part of this. So just be sure you're understanding so far. Just talk to each other about what we did. Make sure you guys are in talking terms with each other. So talk to each other. And then I will erase it. Once you say it's okay, then I will erase it and go to the next step.
Yeah, just like the structure and discuss like what they do.
If there's any confusion, just ask me, okay?
Dr. Boyle,
Huh?
Oh.
so that the flagella protein, does it go all the way down to the kidneys and do it?
Say it again. Actin is right there, right?
The
Axoneme goes down till the ends. That is all axoneme.
Hmm? Not really.
Any doubt here? Any doubt here? Are you still talking?
Are the centriole in the head piece, like the sperm head, or are they in the
Yeah, sperm are, the centriole are kind of where both middle piece and the head, they are kind of joined each other. Any other doubt here? Good, okay, now. So we did talk about what is in the head, right? Now the sperm can travel, right? So when the sperm travel, we call it whipping flagella. Whipping flagella. So flagella whip like this, okay? We call it whipping flagella. Now, the major component of flagellum is axoneme. So axoneme is the major component of flagellum. That's the major component for flagellum. Okay. Now, axoneme is derived from centriole. Okay. Axoneme is derived from centriole. Now, so if you take the axoneme, so this was the axoneme, like we just draw this, right? You cut a cross section of this. You cut a cross section of the axoneme. So the core of axoneme so when you cut it, the core of axoneme have actually structure like this. So in the center, you will have two microtubule cylinders. Okay. And these two microtubule cylinders are surrounded by 9 microtubule cylinders. Okay, now what is there? We all remember from our cell biology. If you don't remember, we said that there is alpha and beta microtubule, right? Those microtubule come together, give a protein called microtubule, right? That is made with alpha and beta microtubule. Those microtubule, they polymerize, give the filament. That filament is called protofilament. So in a way, you'll have like this. So this filament will be formed by multiple microtubule coming together. This will be called protofilament. As I told you on Wednesday, this developmental biology requires knowledge of all the things: genetics, cell biology, biochemistry, all everything, right? So, but I'll keep on reminding you what that is so that you're not confused. But let's see how it goes. Okay. So protofilament. Now what happens? So these protofilament arrange like this: 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Okay? So 13 protofilaments. These are 13 protofilaments. And they're kind of like this, like this, like this, like this, like this. So anyway, you will have a cylinder, right? That is making sense? Okay, like this, our pen has like this cylinder, right? So it will make a cylinder like this, like these fingers are there, going like this. So now, what happens? How these are? So this 13. So this is a circle, right? Full circle of 13 protofilaments. So in the center, right here, you will have one of that circle, and you will have second of that circle. Okay, with 13 protofilament right there. So far it's okay. So this becomes a microtubule. So microtubules are made up of microtubules are made up of 13 protofilaments. And each protofilament is made up of many tubulin proteins. So many tubulin proteins come with protofilament and 13 protofilament. So this is microtubule. That is okay? Now, these two in the center, they are joined by some sort of proteins. So these are connecting. I'll label it in a minute, okay? So these are kind of connecting road. Now what is happening here? So these are two. These two are surrounded by 9. So this is 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay? But these 9 microtubules inside are only 13, but these microtubules which are there, they are actually doublets. So 2 microtubules are joined together. So one is there, with this, there is another join like this, 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 another like this, another like this. So, inside, if you look here, what is inside? Inside are singlet microtubules. 2, these are 2 singlet microtubules. 2 singlet microtubules. Okay? Now outside right here are 9 doublets. 9 doublets. So this is called 9+2 arrangement. Now this arrangement is everywhere there is flagellum, you have 9+2 arrangement. Anywhere there is a cilia, there is a 9+2 arrangement. So if you go up here in test anywhere, you should just remember. Anything that does this thing, that will have 9+2 arrangement, always like this. Now here is the key. If you see here, I have drawn one complete circle, other is half circle. Right? So it means one is complete. One of this doublet this has 13 protofilaments, and this has 11 protofilaments. So far okay? Now, if this is in the cell, part of that cell, right? Flagella, so you should have a membrane also. Right here is this here flagellar membrane, or the sperm membrane. So actually the sperm membrane which was going down, right? But this is the tail piece, so it has become the membrane of flagella, whatever you want to say. So this is the cell membrane now. It's just right here, cell membrane. Or sperm membrane. Now these proteins which are there, they should not be floating here, they should be joined by another protein right here. Oh. So this protein which is joining here, this is called nexin. This is called nexin. Okay. Then there is a protein which joins this outer to inner. So right here is a protein that is joining outer to inner like this. Like this. This protein which is joining there, these are called radial spokes. These are radial spokes. These are again the next type of protein. Now, so when the flagella move, it's like a cylinder. So have you seen the car piston like this? Say go in, go out, right? There's outer core and the piston move like this. So the inner thing is moving like this around the outer, so that allow the things to move, right? So now, here's the thing, we are saying it moves, then you need to have the energy, but before that you should have a motor protein, which can allow these proteins to move, right? So that motor protein is there, and that motor protein is right here. Here. And that motor protein is called dynein. So dynein here have two arms. This is the outer arm of dynein. This is the inner arm of dynein. So what dynein is? Dynein is a motor protein, and it acts as ATPase. It acts as ATPase. What is ATPase and what is motor protein? Okay, so what is motor protein?
It moves along microtubule.
No, it doesn't move. It allows the other thing to move.
Yes.
Motor, like car, right? So of course it moves, but it helps other thing to move on it. What is ATPase?
It hydrolyzes ATP for energy.
Hydrolyzes ATP, so what will happen?
It gives energy out.
Relieves energy out. That energy is released in the form of what? The high energy phosphate bond, right? Phosphate will be released. So, it acts as ATPase. That means it's going to break down the ATP. It's going to utilize the ATP, change that chemical energy into the mechanical energy. Chemical energy change to mechanical energy so that it can move. Okay? And then, this is a critical protein. If this is not working, then what happens? That leads to Hart-Jenner syndrome. Hart-Jenner syndrome. So if the dynein is not working, that leads to a Hart-Jenner syndrome. It's a genetic syndrome. It's a genetic disorder. So what is there? There is non-functional dynein. So what is the result if that is there? Number one, all the male individuals which have this syndrome where dynein cannot work, they will be sterile males. Why?
Can't fertilize an egg.
They can't fertilize egg because sperm cannot move, right? Number two, all of the individual, male or female, irrespective of that, they have very high respiratory infection rate. Very high respiratory infection rate. Why that is so?
Don't you have like Magellan in like the lungs and stuff that help keep like debris and stuff away?
Yes, so there are cilia in our respiratory tract. I just mentioned the cilia, they have the same thing. So the cilia, they basically remove the mucus out of that. So if they cannot move the mucus out, that means you are going to have all those debris, dust and bacteria, everything getting in and accumulating there. Now, most important thing, our heart is on which side? Right side or left side? Left side. In these individuals, there are 50% chances of heart on right side. And how that happen when we go there in the development of heart and all those things will talk. So this individual can have 50% chances their heart can go on the right side. So then, so far everyone is okay here? Okay. Then what happens? We are saying that it needs ATP, right? Where are ATP? ATP is in the middle piece, right? In the middle piece, you have these mitochondria. So mitochondria are in the middle piece. Now, what happened in the mammals? In the mammals, so this was the middle piece, right here the tail starts, right? This was the tail. So in the mammals, what happens? There is a thick sheet right here above here is head, right? So there's a thick sheet like this. There's a thick sheet of fibrous protein. And it's more thick here as it goes down. So more thick here, as it goes down, it tapers. Okay? So there's a thick fibrous, dense fibrous protein there. Dense fibrous protein. And it tapers down as it goes down from the head portion to the tail portion. What it does? It basically prevents the head to whip suddenly, like so it stabilizes the head of the sperm. It kind of does not allow it to whip, otherwise the head will also whipping and can break. So it stabilizes that. So in the middle piece, right here was the axoneme, right? This was the axoneme, so it means mitochondria are making ATP. ATP is being provided to axoneme. Axoneme, dynein is utilizing and helping it like this. But since axoneme is also here, you don't want this portion to whip like this because that can damage the sperm. So that's why you have the dense fibrous protein here, more dense here, going thinner and thinner here, and there is no fibrous protein here. So far it is okay? Now, in mammals, what happened? The sperm differentiation. Sperm differentiation. Is not complete. Is not complete in testes. Is not complete. So although the sperm can move, so sperm can move. It can move, but it cannot fertilize the egg. So sperm has the capability to move, but it cannot fertilize the egg. Therefore, it means it cannot bind to egg. Cannot bind to egg. And that process of further development takes place in the female reproductive tract. Okay? So this activity, which it cannot do, is achieved inside the female reproductive tract. And that process is called capacitation. That is called capacitation. C-A-P-A-C-I-T-A-T-I-O-N. Capacitation.
So capacitation is the sperm not being able to.
Capacitation is the process in which sperm is made ready so that it can bind to egg. So that process, so sperm is not fully differentiated. It can go to the female reproductive tract, but it cannot bind to egg. So that capability to bind to egg is achieved in the female reproductive tract. And that process by which that capability is achieved is called capitation. Okay, so now talk to each other from the structure of the axon intro and make sure that everything is clear so that we can go further. So talk to each other. Make sure you're going through your notes, you're talking to each other.
Did he say can move but can't bind?
So it can't bind until,
But it can move, right?
It can move.
Okay, just checking.
yeah.
Motor filaments.
And does that say next in or next in with the M?
Next, E-X-I-N.
This is structure. Oh, so we're looking at some.
It's the, so it's the like little one that goes through the tail. It's like, it's
So that's what this is.
yeah, that's what that is.
Are you good?
Yeah.
Okay, everything is clear? Okay, explain me, what did you understand?
So you have the protofilaments that form cylinder
Profilament are part of what?
The cylinder microtubules.
Cylinder microtubules are part of what?
The axon.
Axoneme is part of what?
It starts in the middle piece and then goes to the tail.
Axoneme is the part of main component of flagellum, right?
Yeah.
Okay, then what?
And then you have the full ones, which are made of 13, and then the doublet with it's attached and has 11, and you have the 9+2 arrangement.
Okay, then what happens?
Yeah.
Protofilament are made up of what?
Tubulin.
Tubulin. Okay. It's a single molecule of tubulin or multiple?
Multiple.
Multiple, okay. Then what happens?
Then they become microtubules, like you said, in the axoneme. And they're all connected via a few different proteins. Then there's outer and inner dynein heads that use a motor protein to help move the flagellum.
Okay, how
Or the
that
tail
happens?
section.
It breaks them to make energy, like with inducing phase.
Mm-hmm.
And that causes energy that helps it move back and forth.
Okay.
Then you talked about cartilaginous
Mhm.
and then in that's whenever the dynein is not functional. So most of the all the males are sterile. We have a high respiratory infection rate because the cilium
Okay, so this was with the sperm structure, right? So we'll go to now the structure of the egg. I think we will not be able to do it, right? It's already 10 o'clock. So 5 minutes notice of notice. So we can stop it here, right?
Okay.
And we'll keep on doing it on Monday.